Hello, I'm Cristiane Pilon and I'm a peanut physiologist with the University of Georgia at Tifton campus. And today we're going to talk about peanut seed and how important seed quality is. So let's first start with the parts of a seed. A peanut seed consists of two cotyledons and an embryo. The cotyledons contain energy reserves that are used by the embryo to grow into a seedling. Do you know the name of this process? Germination. In the germination process, the embryo uses the reserves from the cotyledons to grow and develop. In the germination process, the seed uses water, oxygen, and the reserves from the cotyledons to grow the embryo. It starts with the radical protrusion, which will be the root system of that plant later on, followed by the plumule growth, which will be the above ground of the plant. In other words, the leaves and stems. When we plant the seeds in the field, we want a fast and uniform emergence of those seeds, right? Speed of germination will depend on the quality of those seeds, and there are many factors that affect that quality. For example, the weather conditions during seed development, or how those seeds are handled and stored after the seeds are harvested from the field, but prior to be planted in the following season. So this is a peanut plant. The peanut plant produces flowers, like this one here, and then at the base of the flower, it will grow a structure called peg, like this one here. So this structure will grow downward, penetrate the soil, and at the tip of the peg, that's where the pod will grow. And this is an example of a peg with a pod being developed. That's where your seeds are. A peanut pod may contain 1 or 2 seeds inside, and they take between 60 to 70 days to get fully mature. But as I mentioned before, there are many factors that may affect seed quality. Drought and temperature stresses are examples of conditions that may decrease seed quality. If a peanut field experiences a drought period, the plants will flower less, and the seeds that are under development will change the quality. Also, after the seeds are harvested from the field, the storage conditions are very important, as high temperature and high relative humidity will decrease greatly the seed quality. But how do we test the seed quality? There are many lab tests that we can do in the seeds to test the quality of them. This is a germination test, and this is the germination paper. I already moistened with water. So here we turn the paper to make it ready for the seeds, to place the seeds. So we're going to move this here and place all the seeds. We're going to use 25 seeds in this case. Okay, now we close with one of the papers. Make sure it's all sealed and roll it. After that, we place the roll in a plastic bag. Seal it, and it's ready to go to the incubator. And this is the incubator. That's where we keep our germination test for 10 days under a temperature of 25 Celsius or 77 Fahrenheit. After 10 days, the germination test will be ready for us to check the seeds. So let's take a look at the seeds here. So when we look at this test and we look at the individual seeds, for example, this seed here, it has a good vigor and germination. The seeds germinated well, the radicle is growing well, and there is some lateral roots growing here. When we look at this seed, for example, it shows a very good vigor. So it's growing even longer radicle, and it has well-developed lateral roots. Let's take a look at this one here. This seed was able to germinate, but the radicle grew a little bit and died. So the vigor is not very good. So this seed will probably not develop into a seedling. So what about this one here? Or this one here? They didn't even germinate. So that indicates that the seed has poor quality. The quality was not good enough to even germinate those seeds. So now that we individually analyze the seeds, we sum the number of seeds that germinated in the test and calculate the germination percentage based on the number of seeds that we used in the test. In this case, we had 87.5% germination, which is a very good germination percentage for a seed lot. But how do we use this? So we know that for this seed lot, if the growers plant those seeds under optimum conditions, we expect approximately 87% germination in a field. Now I have a project for you. I would like for you to run your own germination test at home. For that, you're gonna need a paper plate, some cotton balls, beans, and water. So, the first part, you have to put all the cotton balls over the plate. Put some water. And then place the beans. Let's use 10 beans. After you place the beans, keep the plate in a warm, safe, and dry place for 10 days. Don't forget to take notes on how long each seed will take to germinate. During the test, do you see any difference in germination? Is the germination uniform across all seeds, or does every seed germinate in a different day? After the end of the test, what is the germination percentage of your test? So you'll be able to find out all the answers for the questions and to compare the results you got from what your friends got. So today we learned how we check the quality of the seeds growers plant in a field and how many factors will affect seed quality. For more information about how we connect science and agriculture, please check the other videos at the STEM in Action website. Thank you for watching this video. See you next time.